So thank you for everybody for joining us today. This is an update from Henry Ford Health System on the Moderna vaccine. There we go. On the Moderna Vaccine Trial. Today we have joining us Dr. Paul Kilgore, who's a senior investigational lead for Henry Ford Health System. We also have Dr. Kellie McFarlin, who's a general surgeon at Henry Ford Health System, specializing in minimally invasive abdominal surgery. And we also have Melanie Ivarsson, Ph.D., Chief Development Officer for Moderna. Melanie, if you could share with us. Thank you for joining us today.
Thank you very much for giving me the opportunity to come and talk to you personally. So I'm Melanie Ivarsson, the Chief Development Officer of Moderna. Obviously, you've all seen the very exciting news we were able to share this morning about the interim analysis results of our COVID vaccine trial. And it's a real pleasure to be able to join you today talking to the Henry Ford Health System, because they have really had a very phenomenal role in the conduct of this trial. As one of our highest enrolling sites, we're incredibly grateful to everybody at the site, and in particular the participants who volunteered to be in this incredibly important vaccine trial.
Thank you, Melanie. We appreciate your time, and thank you for saying hello to us today. It was great news released earlier today. So in the fight against COVID-19, we thank you. Now we have Dr. Paul Kilgore, who's a senior investigative lead for the Moderna Vaccine Trial and our COVID-19 vaccine trials at Henry Ford Health System. Dr. Kilgore?
Thank you very much, Tammy. It's great to see everyone here today, and we are very excited about these results. It's really, really encouraging, particularly at this time in the winter when we come up to winter for the pandemic. And one of the very encouraging things is that we're seeing high efficacy for this two-dose vaccine, this two-dose mRNA vaccine. There's a couple more things I'd like to mention. One is that when we think about the results that are out, as Melanie pointed out, these are interim results that we knew were in process. So one of the things that we can expect is additional analysis coming out in the coming weeks, which will be very encouraging. And I think one of the things that you may be aware of is that in the coming weeks as well, the FDA will be talking with Moderna, the manufacturer, to further the development of this vaccine and go through the emergency use authorization application process. And that will take some weeks. And of course, all the data get reviewed very, very carefully. And from our perspective here at Henry Ford and in Detroit, one of the things that we're very, very grateful about is the participation of residents from around the communities in Detroit and Southeast Michigan. It's been a very diverse population that we've been able to enroll in this trial. So that's actually one of the great advantages of the trial. And the support we've received from the community has been outstanding. And it's very encouraging that now we're starting to see these results come to fruition. And one of the things that I think is important to know with the trial and with these results is that we really want to be able to inform communities and the audience and participants who may be thinking about what to do next with the vaccine. And so these results will be very important to look at carefully, but also to communicate widely across the United States and certainly in Southeast Michigan and other parts of Michigan, so people have the latest information on the vaccine. We've had incredible support from the medical community here at Henry Ford. We have an outstanding team of nurses, physician assistants, physicians, laboratory doctors, laboratory scientists, laboratory technicians, and it's really been an incredible partnership with Moderna and with other organizations supporting the trial, including the CRO, PPD, and other groups that really have been instrumental in working with us. And I can't say enough about this team. It's been an outstanding experience.
We appreciate your time, Paul, and appreciate your details. Also, we have with us Dr. Kellie McFarlin. Dr. McFarlin. Dr. McFarlin is a general surgeon. She specializes in minimally invasive abdominal surgery, but she's also a Moderna vaccine trial participant. Thanks, Dr. McFarlin, for joining us.
Thank you.
Can you tell us a little bit about why you joined the Moderna Vaccine Trial and just explain your thought process behind it?
Yes, thanks. I joined the trial for two main reasons. First, as a mom of twin girls, I wanted to do as much as I could to protect my family, and also for my community
You please
to be involved.
all through the talking points. He missed about.
Doctor, we have somebody talking. If you could please mute. Sorry.
Oh, that's okay. Thanks.
Go ahead, Dr. McFarlin.
Okay. Oh, no problem. Thank you. For my community to be involved, to have diversity in the trial was really important to me, to know that the results could be reflective and used for the community that I serve and live in. That was another main reason in participating in the Moderna trial.
Thank you. Do you were you affected at all personally by COVID-19?
Yes, unfortunately, in the very beginning of the pandemic, one of the youngest members of Michigan to pass away was our little cousin Skylar at the age of 5. So we were really pretty devastated by that information that she developed a rare complication from COVID. And that really stuck with our family as reaching out to what we could do to make a difference in our community in fighting the COVID pandemic.
You too have also, I understand, participated yourself in research. Can you explain to us a little bit about why you feel your participation in this trial was important and how the effects of having a diverse population in the research can affect the end result?
Oh, sure. So being a principal investigator for different projects, you understand a lot behind all of the safety checks that goes into a trial. So knowing that even though there's some nervousness in participating really made me comfortable with going into this trial at the phase 3 level, knowing that safety concerns had been addressed and that we were really trying to find out how effective this could be for our community. And again, that gets back to the diversity is if we're not participating in a trial that could directly affect how we do, it's just important to have all of that information available so that I can advise my family, my patients, of what they should do about vaccines.
Thank you. And what is your hope now, knowing the news and hearing the news from this morning?
My hope is that that as the community learns more about how effective this could be, that they would understand that there's safety in this vaccine and would feel comfortable with their physician recommendation of whether they should have a vaccine, and just sort of use this information as empowerment so that they would know all options available.
And can you talk about, people might be apprehensive about the safety or about, taking a vaccine that isn't FDA approved. Can you talk a little bit about your thought about that? Were you nervous?
Yes, I did have some nervousness in the beginning, just because it was new. But as I mentioned before, understanding how studies run and work, I was reassured because every small detail in the study is looked over, whether it's related or not related to the vaccine, to ensure that that it's being done safely. And so knowing that information actually helped me personally, calm down. All my questions were answered by the investigators whenever I had questions. You're free to participate in the trial, and if you're not comfortable, you can stop participating in the trial. There's really no pressure either way. And so just them knowing this open information, all questions answered, really made this a good experience.
Thank you. We really appreciate you sharing this with us, Dr. McFarlin. Dr.
you.
Kilgore, I understand that you have some additional things that you would like to share with folks, some more updates regarding the vaccine trials, that the Moderna trial, and we're also participating in the Johnson & Johnson trial, correct?
That's right. That's right. Right now, we are in what we call the follow-up phase. So we're doing long-term follow-up in the Moderna trial. So we do have participants coming back to our clinic. And during those visits, we talk about their recent experience. We may need to collect a blood sample, and then they're on their way. It's a very quick visit. And in addition to that, we have in another location a new vaccine trial beginning. This is for the Johnson or Janssen vaccine, Johnson & Johnson vaccine. It's a completely different vaccine strategy. Designed the vaccines quite different compared with the Moderna vaccine. As you may know, the Moderna vaccine and the Pfizer vaccine are both mRNA-based vaccines, and those are designed by having a lipid nanoparticle, and the lipid nanoparticle is very instrumental because it contains the mRNA sequence that codes for the spike protein of the coronavirus. And that spike protein is very important because that's how we make antibody to the virus. So the great thing about the mRNA vaccines is that our cell machinery is borrowed by the mRNA to make the spike protein. And as that spike protein is made, it becomes expressed on our cell surface. And from there, with 24/7 immune surveillance, our immune system can begin making the key antibodies that we need to be protected against the natural infection or the wild type virus causing COVID-19. The Janssen vaccine, the Johnson & Johnson vaccine, uses a strategy in which there is a DNA sequence that encodes for the spike protein inserted into the genome of an adenovirus vector. And the difference here is that there's a couple steps needed to make the spike protein. It goes from DNA to mRNA to the protein, but it still really induces an immune response. And what we're seeing in all of these vaccine trials is something very common, and that is that we do see good immune responses to the vaccination. And what that means is that after we're done getting the vaccine, within a few weeks, our bodies start to make that neutralizing antibody that's really critical to be protected against COVID-19. And all of the trial data so far for these vaccines have shown that our bodies do make very good levels of antibody and that are sustained for the weeks after that vaccination is administered. So that's very, very good news. And what we're looking for as we continue the follow-up is just making sure that we can monitor those antibody levels over time. And that's what we're doing right now. For the Johnson & Johnson vaccine trial, as we move forward with the participation, as Dr. McFarlin mentioned, one of the key things that we're seeing already is very good participation from across Southeast Michigan. And this is a very, very important to make sure that we have representation from diverse communities. And that's really a key objective of the Johnson & Johnson trial. It's been a key objective of the Moderna trial and other trials, because we really want to make sure that we have complete representation of all the diverse demographic groups in the United States. And of course, where we are here in Southeast Michigan in Detroit, we're really, really interested to have those individuals participate. And one of the things that Dr. McFarlin mentioned is that the steps for participation is very, very stepwise in the sense that we bring in education for each participant. We walk them through all of the procedures in a very, very step-by-step fashion. They have an informed consent process they go through, and then they complete the enrollment with some sample collection and then administration of the vaccine or placebo. And it's a double-blind, all these studies are double-blinded, which means that I don't know what the participant gets and the participant doesn't know as well. And at the end, the data are being analyzed by independent analysts, biostatisticians, and the data safety monitoring committees. So it's a great partnership, and really the community themselves are key partners in this as well.
Thank you. We also have Melanie Ivarsson from Moderna had another insightful comment she'd like to share.
I just wanted to add on from Dr. Kilgore's point there about the importance of diverse representation in these trials. And in particular, I want to recognize the incredible efforts of the Michigan site in ensuring, particularly for the Moderna study, that we have that diverse representation. Nearly 30% of the participants from this site were of communities of color and of minority populations. And this is incredibly important as we study these vaccines to ensure that they represent the population of the United States, and in particular those communities that have been disproportionately impacted by COVID-19. So really want to recognize, again, the site staff and the participants, and really thank them so much from Moderna, on behalf of Moderna, for their hard work on this trial.
Thank you, Melanie. And thank you, Dr. Kilgore and Dr. McFarlin. I think now we're ready for questions. We have a question from Karen Buffard. Let's see, Karen, can you unmute and ask your question, please?
Sure. I'm wondering if you can provide any idea of the percentage of participants from various communities with regard to their race and ethnicity, and also, did you have any difficulty recruiting diverse populations for the study?
And your question is directed to Dr. Kilgore?
Yes, thank you.
Thank you, Karen.
Well, I'll also suggest Melanie to answer some of this, but just in general terms, one of the things I would say is that with every vaccine trial, as the trial is initiated, one of the key activities that we engage in, along with all our partners, including Henry Ford Health System and all the community members around us, is to get the word out about the trial. And this is very important because it's sometimes true that with other information in the news that it's often difficult to get education out about what's happening with these trials. But today I can tell you that we've had great success in ramping up the enrollment of diverse communities for the Moderna trial, and Melanie will refer to you some percentages exactly overall. For our site in particular, I think also she may want to comment on that in terms of proportion as it represents other proportions in the United States. I would also mention too is that for the Janssen trial and for all the trials, that diversification and participation is absolutely critical. It is a major goal. It's always been one of our goals since we started working on the Moderna trial and now in the Johnson & Johnson trial. And so I think one of the things you're going to be seeing is future comments, analysis about the effectiveness and safety of the vaccine in these populations. So I think that's a great question. Thank you.
Thank you. Melanie, would you like to add?
Yes, of course. Thank you. It was incredibly important, as Dr. Kilgore mentioned there, for Moderna to recruit a study that was representative of the population in the US. And so we, through working very closely with our clinical sites and PPD, our CRO, and through working with outreach to the communities, we actually took the decision about halfway through the trial to really slow down, to ensure that all participants of all different backgrounds were given the appropriate amount of information and the opportunity to enroll in the study. And that meant really focusing on ensuring that our sort of information about our trial was getting to the right community so that people could make an informed decision. And that does take some time, building trust and working out how to engage with different communities. So we ended up with over 10% participants from the African American community and over 20% of the participants from our Hispanic and Latinx community in the study. And in particular, the Henry Ford Health System site did an incredible job recruiting over 9% of the participants that they recruited were from the African American community. Just under 8% were from the Hispanic community, and we had nearly 7% from the Asian community in their trial. So they did a really phenomenal job. We're incredibly grateful for the incredible work that they did. And again, I must always come back to the participants, who we are so incredibly grateful to for entering our trial.
Thank you, Melanie. Appreciate that insight. Does anybody else have any questions for our guest today? I have Gus Burns from MLive. Gus, do you want to go ahead and ask your question? Or would you?
Sure. This is Gus Burns. I'm live, and I had actually a couple. I was wondering if you could kind of talk about the scope as far as how many tests were administered total and how many took place through Henry Ford. And then I think what people are going to be interested in is, if these are approved, how will they be administered? I mean, it sounds like there might be 2 shots involved. Are those taking place in hospitals, et cetera? And how long will they protect people for?
Did you mean how many vaccines were administered? You said tests.
I'm sorry, yes, vaccines.
Potential vaccine study.
So a couple things, and I think Melanie might also want to chime in here. One of the things that I think is important to know is that in the Moderna trial, there were approximately 89 sites across the United States that were enrolling participants. In Michigan, the site in Southeast Michigan here at Henry Ford Health System is the only site in Michigan. And over the course of the enrollment period for the trial, this site here in Southeast Michigan enrolled several hundred participants. And Melanie may have the final number at her fingertips, I'm sure. She's nodding yes. So it was a very, very successful recruitment. And one of the things that I think helped enormously was the ability to communicate word out to the community and diverse populations across Southeast Michigan. The other thing I think to know is that the goal was always to enroll 30,000 nationwide, and half of them approximately received the placebo. And the interim analysis has been done on several thousand participants, but I think what you will also see going forward in the future is more complete analysis, more final analysis on the entire cohort of 30,000. If the vaccine is approved, and knowing what we know from the trials, it is a 2-dose vaccine. And in the trial, we've been administering the vaccine 28 days apart. And the vaccine has a refrigerator storage temperature. It also is stable at some room temperature for a few hours, so it has some major advantages. The Moderna vaccine that I'm speaking of also means that when we administer this vaccine, it's still very important that clinics store the vaccine at the proper temperature. And the way that we do that is we have very specific dedicated refrigerators for only vaccine. And in fact, this is what we do in the trial as well. We monitor the temperature inside the refrigerator very carefully, and immunization clinics around the country that do administer the vaccine will want to do this as well, because maintaining that temperature as recommended by the manufacturer is very, very important to maintain the potency of the vaccine. So that's really important going forward, and that will be for the duration of the entire program in the United States. It really needs to be maintained throughout the program. The other thing I would say finally is that in terms of understanding the duration of antibodies and the levels of antibodies that we will have weeks or several months out after vaccination, we are collecting that data right now. And I think what you're going to see going forward a few months from now and even several months from now is more long-term protection data for the Moderna 2-dose vaccine that will tell us how long people hold on to this antibody. The final thing I would mention is that when we think about antibody protection with the vaccine, one thing I think to keep in mind too is that with this vaccine and other vaccines, we also stimulate a population of T cells. And these T cells are working together with the B cells that make antibody. And the T cells have memory. And when they're talking with your B cells and the immune system, it really is important because those T cells will be able to stick around and then also help you respond if you are challenged by the COVID-19 virus in the future, and you will begin making more antibody. So that's going to be very important going forward for long-term protection as well. So I'll stop there. Over to you, Tammy and Melanie.
Yes, so you said it brilliantly, Dr. Kilgore. Thank you very much. I would just like to add that the Michigan site actually enrolled over 700 participants for our trial. They were one of the highest enrolling sites in the country. So again, thank you for the incredible work this site has done.
Thank you. I'd also like to thank our media friends for sharing the message about the recruitment being available, because they were integral to getting the message out too. So, we had another couple questions here in the chat. Karen Bouchard from the Detroit News asked, can you explain in layman's terms why the Moderna vaccine doesn't need to be stored in ultra cold temperatures as the Pfizer vaccine requires? Paul?
Here again, also Melanie may want to chime in. Melanie, I'll let you go first, and then I'll check.
It's related to our tech dev and our formulation, and how we have been able to achieve stability. And we have been able to create a vaccine that is stable at refrigerated temperatures. It's probably not appropriate right now to go into all the technical details, but the stability data has been worked through by our tech dev colleagues, and they've just been able to create the formulation so that it's stable. So with vaccines, you tend to sort of improve the formulations over time. We're fortunate to have got a really fantastic formulation team that have been able to conduct that very careful stability data to show that it's stable at less frozen temperatures.
That's great. And I would just add in too, in the trials that have been done for the Pfizer and Moderna vaccines, one of the things that you know is that they are both formulating their vaccine using the lipid nanoparticle that I mentioned. And the mRNA is encased by the lipid nanoparticle. So the mRNA is kept stable with a lipid nanoparticle. So the structure of the lipid nanoparticle itself is actually very important. And in fact, even though I use the term lipid nanoparticle, the particle that the Pfizer vaccine uses may be slightly different compared with the particle that the Moderna vaccine is using. The second reason why there may be a difference in the storage and the stability is that the concentration or the content of the mRNA in the Moderna vaccine is 100 micrograms, and that's in the phase 3 trial. In the phase 3 trial for Pfizer, the concentration of the mRNA is lower, around 30 micrograms. And that may also mean when you have higher concentrations in a lipid nanoparticle, you have more mRNA that stays around even with a higher temperature. So because there's only 30 micrograms in the Pfizer mRNA vaccine, it's really critical that those 30 micrograms remain intact and able to transform to produce spike protein that I talked about earlier. So it's likely related to a couple factors at least. One is the structure of the lipid nanoparticle, and number two is the concentration of the vaccine antigen within the particle itself that makes these vaccines different.
Thank
I hope
you.
that answered your question.
Thank you. And Erica with Fox 2 asked, how soon could this be available widespread in estimation? Melanie, that looks like a question for you.
It does, doesn't it? Thank you. So yes, we will now start preparing all the documentation for submission, as Dr. Kilgore outlined earlier, for emergency use authorization. And you will have seen that the FDA are working, publicly waiting for us to provide that data. They're going to review it very carefully. They're going to perform an independent review of the data package that we provide. So we're going to provide the safety and the efficacy data to them, as well as all the information around the manufacture of our vaccine. And they're going to assess that and independently review that very, very, very carefully. And if they agree that it's appropriate, they will move forward to emergency use authorization. We are manufacturing our vaccine now so that we do have doses available in the month of December. So it's really going to be critically important that we get our documentation together, provide that for independent review, and that if an EUA application is approved, then we would be able to release vaccine in the month of December.
Okay, thank you. And Melanie, where are we speaking to you from? Or where are you located?
I'm in Boston. Our headquarters are in Cambridge, in Boston, and we're all working from home, obviously. So I'm just outside Boston, Massachusetts.
Okay, thank you. We had a media person asking location. And Dr. Kilgore and Dr. McFarlin, you're both at Henry Ford Hospital in Detroit, correct? Mm-hmm. Okay. Lisa RC asked, the findings have yet to be peer reviewed. Is there an ETA on when that will occur?
Yeah, as we put in our press release yesterday, I believe, that will, as the full analysis is available, this is an interim analysis, and as a full analysis is made available, absolutely that will be peer reviewed. And then as the data is pulled together, that will go to the FDA. And then obviously there is the VRBPAC process, which is the Vaccines and Related Biologics Advisory Committee, that will also look at this data as part of that process.
Thank you. I don't see any other questions. I think we're all set.
Sorry, this is Will Callan with Michigan Radio. I do just have one question, if that's okay.
Sure.
I just wanted to ask, I know that for both this Moderna vaccine and the Pfizer vaccine, both are using this mRNA technology, which is relatively new. Are you at all surprised by how effective this new technology is? Can you speak to that a little bit?
And your question is directed toward?
Whoever can best answer it, but I suspect Dr. Kilgore and Melanie.
That's an interesting question. I'll give a brief answer. It turns out that the mRNA technology has been an active area of vaccine research for a good 10 to 15 years. What's interesting in the world of vaccine research is that sometimes a lot of work kind of flies under the radar until it really grabs attention. And Moderna and the team in Moderna, and Melanie can probably give even more precision on this, have been working on a whole host of vaccine candidates. In fact, before COVID-19 broke out, they were working on a flu vaccine using the mRNA technology. And I believe they still may have that. They also were working on other vaccines for CMV and human metapneumovirus. So it had been in the works for a long time. I guess I'm not surprised because before the mRNA technology had arisen, there were many groups working on what's called a DNA vaccination. DNA vaccination was a strategy that was devised several years ago as well, had been worked on. It had not experienced as much success as the mRNA approach. And one of the reasons why the mRNA technology in this vaccine may be working so well is that the mRNA is encased in that lipid nanoparticle. And it's a lipid nanoparticle which is really critical, I think, because it helps fuse, the lipid nanoparticle, in fact, will fuse with the membrane of a cell in your body. And it's really that fusion that allows the mRNA to enter your cell and then work with the machinery in the cell, like the endoplasmic reticulum, the ribosomes, the Golgi apparatus, to make that spike protein. So I guess the short answer is I'm not really surprised that it's working. And it had been flying under the radar, but now with COVID-19, it's really becoming well recognized, I think.
Thank you for that answer, Dr. Kilgore. Appreciate it. Let's see. Any other questions out there? And with that, I want to thank you, Dr. Kilgore, Dr. McFarlin, and Melanie for sharing this information. And if anybody in our media team, media colleagues you could share the link for the sign up for the J&J trial and media that you do. That's henryford.com/J and spelled out J vaccine. Thank you, everyone. Have a great day.